Welcome to the Wednesday afternoon lecture. A very impressive opportunity to learn about anesthesia and what it tells us about consciousness and the human brain, one of those very fundamental questions that all of us have hoped to get answers to. And we have a spectacular presenter today, Emery Brown, who I'll introduce in a moment. Let me just say a quick word about this particular lecture, which is named for DeWitt Stetton, Jr. This lecture was established in 1982, almost 40 years ago, to honor DeWitt Stetton. He was the third director of NIGMS and oversaw some very significant growth and development of that institute during his time. He was particularly devoted to support of basic science, including genetics and cellular and molecular biology and chemistry. He was particularly well known as a co-author of the classic text, Principles of Biochemistry. And so he was honored by the establishment of this annual lecture, and we're delighted to remember him on this occasion. Our speaker today, Dr. Emery Brown, is currently the Edward Hood Taplin Professor of Medical Engineering and of Computational Neuroscience at the Massachusetts Institute of Technology, but that's not enough. He is also the Warren Zeppel Professor of Anesthesia at Harvard Medical School and Massachusetts General Hospital. I don't know that there's anybody else in the world that has quite this same combination of titles and skills. He's a Harvard guy, undergrad at Harvard, MD, PhD at Harvard. His PhD was in statistics. He then went on to internship in internal medicine at Brigham and Women's Hospital and a residency in anesthesiology at Harvard Medical School, Mass General Hospital. His research over the years has been of the highest quality and has been recognized in multiple ways, including being elected to all three of the national academies. And you won't meet very many people like that. So he's a member of the National Academy of Sciences, the National Academy of Medicine, and the National Academy of Engineering. He won the Dixon Prize in Science in 2018. He's had a Pioneer Award. I'm going to give you some things we're proud of from NIH, as well as a Transformative Research Award from NIH. He's ably served us in various capacities on the Council of Councils, and I particularly want to thank him for his service as part of the Brain Initiative Working Group, where his input has been really important in shaping this very bold program to figure out exactly how the circuits in the brain do what they do. His research is basically what you'd call systems neuroscience, trying to study how the mechanism of general anesthesia induces loss of consciousness and what that tells us about the function of the human brain. A very profound set of questions indeed. His topic today is Rethinking General Anesthesia. We're fortunate indeed to have him with us, speaking to us remotely and virtually from his home, but he's going to tell us some pretty interesting stuff. So without further ado, please virtually welcome Dr. Emery Brown.
Francis, thank you so much for that great introduction. John, thanks so much for the invitation to speak here today. I've entitled this Rethinking General Anesthesia, and I'll go through my outline, but before I do, I want to just go ahead and acknowledge the very generous funding support that I've had, particularly from the NIH. As Francis mentioned, I had a Pioneer Award. That's where I actually got really started in earnest in 2007 doing this research, and followed up with a Transformative Research Award. And my colleague, Patrick Purdon, had an NIH New Innovator Award, and we currently have the good fortune to be supported by an NIH PO1. All of this is from NIGMS. And we've had, very generous funding support from the Department of Anesthesia and my three institutes, which I'm affiliated with at MIT. I have one or two disclosures here. Massimo has licensed IP based on some of the work I'm going to present here today. I do hold founder shares in like a new startup called Pascal. So just so we're all upfront about that. So let me just push ahead to what I'm going to talk about. I'm going to follow this outline. I'll give you a quick clinical look at what general anesthesia is. I'm going to talk specifically about loss of consciousness induced by propofol. And then I'll give you just a quick summary of some information, how dynamics in the brain change with class of drug, some of the mechanisms, and something about patient age, which is a key factor in thinking about taking care of patients clinically. And then all this is building up to the idea of how can we improve what we do in taking care of patients under anesthesia. And I'm going to talk to you about this idea of multimodal general anesthesia, which is something which we've been practicing for the last few years or so, using more than one drug simultaneously to better create the anesthetic state. And then it's all about really controlling the brain. So I'm going to show you some work we've done on closed loop control of anesthesia. And then anesthesia up until now is about turning the brain off, keeping it off so you can have surgery. I'm going to tell you a little bit about reanimation. It's turning it back on. So here are the main points that I'm going to make. One of the, it's often said it's not clear how anesthesia works, and we think that nothing could be further from the truth. I'm going to show you that in just a few seconds. Anesthetics work by creating oscillations, and the oscillations alter arousal. They induce oscillations, and these oscillations, I'm going to show you based on recent non-human primate work, modulate very dramatically the spiking activity of neurons across the entire brain. These oscillations are very easy to see in the EEG. They're very strong, and they change systematically with the drug, with the patient age, and also, this is, as we're going to see in a sec, allows us to design control strategies for maintaining an anesthetic state, a desired anesthetic state. And I'm going to show you a bit about this multimodal anesthesia idea. And the idea here is that you can target multiple pathways in the nervous system that control pain or nociception. And by so doing, you can reduce the amount of drug you need to make someone unconscious. So you win by not giving someone as much anesthesia to keep them unconscious, and you get essentially better control of nociception and pain. And clearly, this is one of the ideas which we're studying to help reduce the use of opioids. And then finally, reanimation, turn the brain back on. And we think that this is an idea which can help reduce the cognitive dysfunction that's often seen, particularly in elderly patients after anesthesia. So what is general anesthesia? Well, it's this drug-induced state. It's reversible. It consists of anti-nociception, meaning control of the nociceptive processing, unconsciousness, amnesia, akinesia. We do that while we maintain stability and control of the physiologic systems, and it's reversible. We bring you in, we bring you out, and it's drug-induced and drug-maintained, I should say also. I started this here because when we wrote this original piece in New England Journal back in 10 years ago now, we didn't say anti-nociception. And I'm going to be talking about nociception to distinguish that from analgesia. So analgesia is anti-nociception and analgesia. Pain is basically conscious perception of something that's a nociceptive signal. Whereas if you're unconscious, it's just nociception. The nociceptive pathways or the pain pathways can be active. So under anesthesia, when you're unconscious, what you technically have is anti-nociception. And that's going to be important when I talk about the multimodal anesthesia. So that's our definition. So let me show you what anesthesia looks like clinically. So in all of our ORs, we have EEG, electrode, EEG setup, so we can monitor the brain states of the patients having anesthesia. And I'm going to play a little video that I made about 7 years ago on a woman who was just about to go off under anesthesia. She's 60 years old. She weighs about 80 kilograms, and I'm going to give her 150 milligrams of propofol. And I'm going to inject it, and then we're just going to watch how her EEG changes. So you can see the 4 leads here. So there are 2 on the left, and then the 2 on the right. So I'm just going to play this little video here. And what you're going to see here in a second, this is going to get really noisy. Those are eye blinks, right? It's going to get real noisy, starting about right here. And the reason it gets noisy like that is that when you inject propofol into a small vein, like one on the back of your hand, it burns like crazy. So you see all this noise that's occurring here. That's her tensing up because the propofol is burning. Now, that's actually going to be good from a pedagogical standpoint because you're going to be able to see exactly when the anesthetic is another 5 seconds to watch this. So right here, watch this. You're going to see beta oscillations. Okay. Now watch these large slow oscillations come on. That's your brainstem being inactivated. They're going to get deeper. And now watch, she's going to go flat, reverse, flat, Yeah. So what did I just show you? So this was, here she was awake up here where you saw this is about five microvolts here. And then with the advent of with the induction, you saw these beta oscillations appear here. That's when these oscillations about 12 to 16 cycles per second. And so if I were trying to keep her unconscious just for a colonoscopy or what have you, this is where I would try to maintain her with oscillations like this. This represents a sedative state, all right? So 12 to 16 cycles per second. And then she went from there, from C right down to E, these large sort of slow oscillations. And just as a rule of thumb, we can talk about the mechanism in the Q&A. This is like usually brainstem inactivation. So this is precisely if you were to have been in the operating room with her, when these came on, you would see when she lost consciousness. Then she went into this very deep state of anesthesia called burst suppression. She just passed right down to here. And between the bursts, she was isoelectric, she was flat. You saw she was flat and then bursting. You didn't see this state here at all, the alpha oscillation riding on top of the slow oscillation. So this is like about 10 cycles per second. This is the classic pattern for propofol, is what we're going to talk most about. And so you can see what you do when you go from in a state of anesthesia, you go from being in these low amplitude, sort of high frequency oscillations to these very low frequency, high amplitude oscillations like you're seeing here. And one of the main points I want to make here is that this is the strongest signal, strongest EEG signal there is of all the things that the EEG is used for, whether it's to track patients who understand sleep states, cognitive studies, you're examining patients and trying to evaluate the brain state of someone who's in a coma, or maybe using the EEG for biofeedback. There's always noise. And you saw what happened here. There was noise on her EEG, but as soon as the drug took over, you got these very clean signals. And you're getting clean signals also because large numbers of circuits in the brain are starting to do exactly the same thing. I'm going to be able to show you that very directly in just a few minutes. So it's ironic that this is the strongest EEG signal around, probably has the most information about the brain state that it's tracking, in this case anesthesia. The anesthesiologists are probably the least likely to use it. So the way I'm going to show the data going forward is going to be in this format, going in what's called the spectrogram. So looking at this in time here, frequency here, and then the color coding is going to tell us something about the amplitude of the oscillation, and the location on the frequency band here is going to tell us something about the frequencies that are present here. So this is a young woman I took care of a few years back. She was having a small procedure, so I gave her a bolus of propofol to induce, and I started an infusion of 100 micrograms per kilogram per minute. So keep the number in mind, 100 here. We're going to see it, another infusion later on, which is going to be a lot less. So she got the bolus of propofol here, near the large slow oscillations come on, and you can see now they're the slow delta oscillations. So going between about 0.1 to 4 hertz, and up here going between about 8 to let's say about 10 to 12 hertz here. So this is that alpha oscillation. And you can see it up here in this particular one. This one here is just slow. So it's just, just going up and down. But now you have the higher frequency component on it too, which is the alpha oscillation. So you can see both of them here. So as long as you run this propofol infusion, you keep her brain in this state. You have the slow oscillation together with the alpha oscillation. When you turn it off, and a few minutes later, like over here at about 32 minutes, she woke up. So this is classic propofol. If you give propofol to a young person, 18 to 35, let's say, for the sake of this conversation, you're going to see a pattern like this. And one of the things that we spent our time doing in the last few years is really trying to understand this pattern and what is its relationship to someone being unconscious, all right? And so these are the colleagues that I've had the good fortune to work with on this particular project. So my longstanding colleague, Patrick Purton at Mass General Hospital, neuroscientist, bioengineer, Nancy Coppell, mathematician at Boston University, expert in dynamical systems, Chinang Cheng, former postdoc, who's now at WashU, Ahmad Eskandar, neurosurgeon, now at Einstein, Eric Pierce, anesthesiologist at MGH, Laura Lewis, former PhD student, who's now professor of biomedical engineering at Boston University, and Sid Cash, our neurology colleague at Mass General Hospital. So I want to show you one other bit of phenomenology, then we'll talk mechanism. Back in 1977, John Mischenfeld and John Tinker did a series of experiments, anesthesia experiments in monkeys in which they recorded EEG. And they named this phenomenon called anteriorization. And what anteriorization is the movement of oscillations from the back of the head to the front of the head when the animal became anesthetized. So I just showed you that there's a very rich temporal dynamic in the EEG when someone's anesthetized. There's also a very rich spatial dynamic as well. And so what this is these are 44 sites, 44 of 64 sites on the head of one of our study subjects, one of our volunteers, that we gave propofol to in increasing doses and decreasing doses, so we could study the whole spatial dynamic of the oscillations by measuring EEG at the same time that he's receiving the propofol. And so he's got his eyes closed here. So this is the front of his nose is up here, left ear's over here, right ear's over here. And in each site, each one of these 44 sites, I'm showing you the spectrogram. So this is him before we started. He's lying there with his eyes closed. And what you may remember is if you close your eyes and you measure the EEG in the back of the head back here, you're going to see a 10 hertz oscillation. And pretty much everybody has it. Maybe it gets a little bit weaker as we get older. Maybe about 5 to 10% of people don't have it. But it's a very well-known phenomenon. It's called the eyes closed alpha oscillation. And we ask our study subjects to keep their eyes closed so that we don't get like blink artifacts on EEG, or at least we minimize it. So what you're going to see is as I give him increasing doses of propofol, we're going to go up 5 different levels, each of which is going to last for about 14 minutes, and we're going to come down. You're going to see this alpha oscillation, which is here in the back, move to the front, concentrate here in the front. It's going to be very coherent across the front part of the scalp here. And then when we turn the propofol off and let him recover, it's going to move back this way. So that's the phenomenon of anteriorization that Tinker and Mitchenfelder identified back in 1977 in monkeys. I want to show you what it looks like. I'm going to show you what it looks like in humans. So I'll play this. So baseline, there's no drug administered yet. Now the infusion starts. And watch how the alpha oscillation breaks up in the back. You see it appearing in the front. And it gets to be very intense. And we're only up to level 3. And it's coherent across the front part of the scalp there. And level 5 here, now it's going to be very intense. Now watch what happens when we back it back, when we turn it off, and reduce the level, reduce the infusion rate. It moves backwards and reappears in the back of the head back here. And if we let it continue, we turn it off and give it a few more minutes, he comes to. So that's anteriorization. So what's going on there? So I can draw a diagram like this and talk about where propofol acts. So propofol works through GABAergic circuits. It enhances GABAergic inhibition, and GABAergic interneurons are everywhere. Cortex here in the thalamus, in the brainstem, all the way out through along the spinal cord as well. So everywhere that you have GABAergic interneurons, propofol is very lipophilic, so it goes to all these sites. I can draw a diagram like this, and we drew diagrams like this, and then you go to the operating room, you see this. So how does this relate to this? Okay, that's what I want to try to explain to you now. And then look here, you have the alpha oscillations, which I just showed you, which they concentrate in the front of the scalp here when you're profoundly unconscious. And the one thing which I mentioned, but I didn't really emphasize, is that this alpha oscillation is coherent. So all of these sites are across the front of the head here are moving in unison at the same frequency, at the same phase. So it's like locked essentially. So let me just unpack this a little bit. What's going on here? So let's talk about the alpha oscillation. So one of the things that this is why my collaboration with Nancy Gopel is so important. When we first identified this, we realized that it was most likely due to the actions of a central source. And so through the modeling work that we did with Nancy, we postulated that this is a thalamocortical oscillation, a rhythm going back between the thalamus and cortex. And there was good physiologic reason to think that it was very easy to drive the cortex to an alpha oscillation. Our modeling studies showed, and at the same time driving the thalamus to one. And the two oscillations essentially lock, is what our modeling studies told us. So we subsequently went in rodents, and also now in non-human primates, stuck electrodes here in the thalamus and here in the cortex, and actually verified that. So that's part A. you've got this alpha oscillation here, which is very, very strong. It looks like it's tying up the thalamus and cortex. And so just that alone, if you think about it, remember the thalamus is a very important way station. All sorts of information goes through there: visual information, auditory information, somatosensory information, pain information. So if you tie up the thalamus, you're going to really tie up a major sort of, like I said, way station. So the idea that you could possibly be unconscious by doing that is very, very plausible. But that's not the entire story. Because look at this guy here. Look at these slow and delta oscillations. They're just as powerful. So they must be playing some role as well. And they certainly are. And so our first insights that we got into this came from our studies in patients that have epilepsy that have electrodes implanted. So these are patients who have intractable epilepsy. They need surgery. They come in, they failed medications. They come in, they get electrodes implanted to identify their seizure foci. And Sid Cash and Ahmad were conducting research as well. So they often place, several research electrodes. So we're able to record neural activity, not only field potentials, but spiking activity of human neurons directly from the human brain. And this is one of the big issues. One of the things, the most exciting aspect that the Brain Initiative is supporting, particularly this type of research, because this is a chance to see what neurons in humans do in various conditions. So these patients have been very important, not only to our science, but also the science of many other people. So once they've had the electrodes implanted, they stay in the hospital for 5 to 7 days. They seize off of medications, and then the care team figures out where the seizure focus is. They come back in to have the electrodes removed, and a subsequent visit to the OR, they'll have their resection. So just look, so these are field potentials here that are recorded at the 3 sites here on the brain, all right? And then here's what the neurons are doing. The neuron is pretty much spiking pretty much wherever it wants to. That's a neuron which is next to the red electrode, all right? Now look what happens once the slow oscillations start. Once the slow oscillations start, that induced by the propofol, you can see the neuron spikes here, spikes here, spikes here, spikes here. So it's spiking roughly about every 2 seconds or so, right? So its spiking is great. The frequency with which it can spike is actually greatly reduced. And so if it has to spike sort of in synchrony with neurons in locations nearby, it's going to be very hard to do. And you can start to see why someone would be unconscious. But let's drill a little bit deeper, because we have new information on this now from our non-human primate studies. So this is work that we've been doing, as I mentioned, thanks to our NIGMS P01 with Earl Miller, and also Jake Donahue, who's an MD PhD student who just finished his PhD with us. But here's the same thing in a non-human primate who's anesthetized with propofol. So this is the frontal eye field. So this is a frontal brain region, 2 frontal brain regions, sort of the posterior parietal area, and also the superior temporal gyrus, sort of over the auditory area. So each one of these, so the little dots, each row is one neuron, and the color coding shows you the neurons that are coming from the different brain regions. So we have about somewhere between 10 to 20 neurons we're showing you here from each of these regions, and you can look at them over time. And this is once the animal's anesthetized. So you can see there are these long swatches of time right here. You can actually see the modulation by the slow oscillation, and how there are times that things are well in phase, but when they're not they're well in phase, but then there are large periods of times where they're shut down. So you've got this sort of fragmenting, if you would, of the cortex. And just to show you, give you another shot at this. So this is what the animal would look like when it's awake. So there are the field potentials in these same areas. This is what the spiking activity in the brain looks like. And here's the spiking activity now that the animal is unconscious. And so you can see the slow oscillations are strongly modulating when these neurons spike, right? And to give you some quantitative feel for how much of a change this is, let's say from here, the awake state, to the unconscious state, this is some analyses that Josefina Correa has just done for us as part of her PhD work. So if you look here, so these are those same areas. This is PFC. 8A is the frontal eye field, superior temporal gyrus here, and the posterior parietal cortex. So this is spike rate on the y-axis in each of these areas, and this is time here. So this is the point where the drug infusion was started here. So the animal's awake over here. And I just want you to look at the scales here. So this is somewhere the spike rate, so the number of spikes per second, is somewhere between about, let's say, 7 to 10, 7 to 11 spikes per second. The propofol runs in, the animal becomes unconscious right here, and the spike rates drop profoundly. So this goes from about, as I said, 7 to 10 or 7 to 11 spikes per second to about a half to 1 spike per second. So there's a dramatic drop in spiking activity that's also modulated. So why do we care about this? All right. So we care about this because of what I was saying before. If you go back here to the operating room, you can see this in the operating room. What I've been describing, I just described in detail what we think is happening during this slow oscillation here. And, you hear about anesthesia, not because people go, wow, anesthesia is a really cool subspecialty. They're doing great science work. Isn't that really exciting? No, you hear about it when we screw up, or at least we're alleged to screw up. And people write articles like about, oh, wow, there was another case of someone being, under anesthesia, but they turned out they were awake and the anesthesiologist didn't realize it. So awareness, right? Awareness should never happen now, because if anything that I've said is even remotely correct, if what I said about the alpha oscillation is correct, what I said about the slow oscillation is correct, then it's pretty clear to us that you can know in a very informed way whether someone was unconscious. Or let me put it this way: if someone came to me and said, "Emery, look, I had anesthesia, last year, and I was, I had a case of awareness," and it said, I'm coming back to the OR, and say, "Look, I'll take care of you. Here's what we'll do: we'll use the EEG." And I would dial the person down until I saw oscillations like this. And like I said, if what I said about these thalamocortical circuit being tied up, or the slow oscillations, as you see them strongly modulating neural activity, so there's very little spiking activity, so it's extremely difficult for the parts of the brain to communicate, then I'd be very confident that the person is not going to have awareness. The difficulty is that based on our estimates, only about 23 to 25% of anesthesiologists use the EEG. And those that are using it are not using it the way I'm suggesting here. So just to push a bit further, I've shown you, we did a little bit of a deep dive into propofol. But now here are the other drugs. So sevoflurane, which is still an ether. Remember, ether was the first anesthetic that was discovered back in the 1840s by Crawford Long in Atlanta, and also by John 1842, and William Morton in 1846 at Mass General Hospital. Well, if you look at them like this, maybe you can see that there are differences in these patterns, but the one thing which we've been pushing is using the spectrogram in real time in the operating room to actually see the differences. So you can tell the difference in like the drug dose, the drug class, and also, as I'm going to show you in a second, like patient age. And just like I've done for propofol, I won't go back over these other drugs in the same degree of detail. We can drill down and have a detailed mechanistic discussion about how the oscillations are produced based on where the drugs are acting in the brain. And the same way that we've done with propofol, we're doing this now with the other drugs as well. And so this framework of thinking in terms of brain circuits, where the drugs bind in the brain to which receptors, and how that alters circuit dynamics, we think is a key element for understanding how the drugs work. But the real important one of the important payoffs of that is that it also tells you how you can infer whether someone's unconscious and under anesthesia in the operating room, which is obviously of utmost importance clinically. And the final sort of background point that I want to make is that the brain's response to anesthesia changes dramatically with age. So here's that someone who's around 30 years of age. This is like the 19-year-old woman I showed you. This is all propofol. So here's the alpha oscillation. There's the slow delta oscillation. Here's someone who's 57. You can see the oscillation is there, but it's a little weaker perhaps, right? Here's a lady over here who's 81. The oscillations are much weaker now. And you can see both the alpha oscillation as well as the slow oscillation. And this is the lady I took care of she had a tumor on her chest the size of an American football. And it took the thoracic surgeons the better part of 6 hours to remove it. And I was able to give her 1/3 of the already age-adjusted dose of the drug infusions because I was watching her EEG in real time, was able to wake her up on a dime. So here's the part which is a little bit interesting. These two guys are roughly the same age. His EEG looks like his, whereas his EEG looks like hers. And you think, oh, well, it seems like it's surprising at the moment. But if you forget about, well, there are two things. One is we age different physically, so it makes sense that our brains probably age differently as well. And then the other thing is that if you forget about anesthesia for the purposes of putting someone in a state so they can have surgery, think of it as an experiment where you're putting in a stimulus. What the stimulus does is it makes the brain circuits oscillate. So when you're younger, let's say in your 30s, you can oscillate like this, but as you get older and your circuits start to break down, and not break down necessarily with, you don't have to argue for dementia or Alzheimer's or that sort of thing, because like in this case, the neurons here have been around for 81 years. They've been around for 50 some years in the case of this guy. So the myelin breaks down, the mitochondria don't work as well, the dendrites don't extend and retract as well, you don't produce as much neurotransmitter. So it makes sense that you'd have a diminished signal, but the rate at which it diminishes probably also depends very much on how well we take care of ourselves. So, and that's probably what we're seeing here. And this is something which we're going to start looking into. But, and you hear the kids, I mean, when you see the spectrograms of the kids in the OR, I mean, they're actually really gorgeous. They look like, I mean, you know, look like abstract art. But you can see, if you look at it this way, you can see there's the same dynamic across all these age groups, but the width of the spectral bands are different depending upon the age. And so actually the power, it turns out we've learned empirically, is largest between the ages of 6 and 8 years. And after that, it sort of declines, you know, sort of monotonically. But, you know, so what's very clear is you need a different monitoring strategy, which is very easy to see for, you know, for the young kids compared to, you know, like older adults. So now let's turn this into something that we use in the operating room. So this is my colleague, Marusa Naranjo, who's from Merida, the Yucatan Peninsula in Mexico. We've been working on this project for about 4 or 5 years now, and we call it multimodal general anesthesia. So here's the idea. So this is how anesthesia is typically done. We maintain the various drugs, maintain the various states by giving drugs from different categories. So unconsciousness, we'd induce, like I said, with propofol, you'd maintain the unconsciousness with an inhaled drug like sevoflurane, which is an ether. And, you know, for analgesia, you might principally use opioids. You know, if someone is unconscious, you don't need to give anything explicitly for amnesia, but, you know, you can imagine giving, you know, benzodiazepine maybe before the surgery or that sort of thing. But if someone's unconscious, that's not necessary. Then you have special drugs for which we give that block the neuromuscular, the activity of the neuromuscular junction, anticholinergic drugs, which induce mobility or what we call muscle relaxation, or is colloquially referred to as paralysis. Then you give drugs to maintain hemodynamic stability. Just to give you some idea, just the basic anesthetic, someone's having a, let's say their appendix removed, you're going to get at least 10 drugs. The only drug here that does everything, you can see the inhaled drugs, these are the ethers. So it's, you know, there's no surprise that, you know, they're the ones that were, you know, sort of first discovered, because you can get pretty much everything with a single drug. And the whole idea behind balanced anesthesia is to sort of divide up the effects. So by giving small doses of each drug, each of the drug or drugs from different categories, you can get most of the good effects and fewer of the side effects. So having said that, you know, this can be optimized further, and this is what we call multimodal general anesthesia. And so, and it's based on a very, very simple idea, and it's in this first statement here. You need anesthesia because it hurts. Surgery hurts. And so what we should be doing is coming up with multiple ways to target the nociceptive system or the pain system. So why not use multiple drugs, not just multiple drugs in general, as I was suggesting before, but specifically multiple drugs to target the nociceptive system. Now, by doing that, it turns out that those drugs also decrease arousal. So what that means is the amount of propofol or sevoflurane that you have to give to keep someone unconscious becomes a lot less. I mean, this is especially important for older patients. You have to be able to monitor arousal and have a monitor of antinociception. So here, this is going to be the EEG. And here we're going to use heart rate and blood pressure. And then you should continue your multimodal strategy into the postoperative period. So I'm just going to show you an example of this in a woman that we took care of about 2 years ago. She's 89 plus, and as you know, anytime someone's over 89 years of age, you can't say how old she was, but let's just say she was plus, as it indicates here. She had a history of diabetes and high blood pressure, and she was coming for a laparoscopic right colectomy. She's about 5 feet tall. She weighs about 57 kilograms. So here's my tri-drug regimen to control nociception during the surgery. I use dexmedetomidine, this alpha-2 adrenergic agonist, remifentanil, opioid antagonist, and then the opioid, I should say, and then lidocaine, infusion of lidocaine. And then, so the payoff of this by using, alpha-2 circuits, opioid circuits, and then lidocaine, which we'll discuss in a second, like what the mechanism is, but it's a very useful antinociceptive adjunct. It's well documented. I was able to keep this woman unconscious with just 30 mics per kilo per minute of propofol. And I just remember I said before, that 100 micrograms per kilogram per minute that I was giving to the younger woman. This is next to nothing. I mean, this is like a homeopathic dose of propofol, but she's profoundly unconscious, as I'll show you in a second. And we use rocuronium for muscle relaxation and oral Tylenol for post-op pain control, as well as a bit of IV hydromorphone. Right. So these are her vital signs. Her vital signs are quite stable during the case. And you can see from her, she has just really slow oscillations. And I just wanted to show you this because this is very typical. It's like what I was showing with the woman who was 81 years of age. Once patients become older, all you see are just sort of low frequency oscillations. The ability to produce this alpha oscillation has been lost. And this is just an empirical observation. I can make conjectures as to why that's the case. But empirically, all you'll see is just essentially slow oscillations. And again, we had great control of her anesthetic state with literally next to nothing. I mean, there's no way in the world someone would just you would guess that you could just give this woman 30 mics per kilo per minute of propofol to keep her unconscious. But the reason we're able to do it is because we're monitoring the EEG. We can see that she is profoundly unconscious based on the presence of these slow oscillations, like I was showing you that were there in the monkey and also in our epilepsy subjects. And we can use that information to be confident that she's unconscious. And so what's happening here is, here's the idea. You have a drug, let's say like the opioids or ketamine, which you might use for antinociception, or dexmedetomidine. So if you take ketamine here, so here's one of the key points where it has its antinociceptive effect at this first synapse that comes into the spinal cord here. But if you look at where else it goes, this is an NMDA antagonist. So you have projections coming out of the brainstem going to central thalamus to basal forebrain, which are glutamatergic NMDA pathways. So it's actually going to have effects here. So this is primarily a nociceptive effect here. This is a decrease in arousal. So each one of these drugs has a very similar sort of profile. So now, as a consequence, because you're taking advantage of this secondary effect on arousal, you can reduce the propofol that you need to keep someone unconscious. And what we've seen is we're able to wake people up on a dime with much clearer heads. And this is what I was about to say about lidocaine. We think there are two mechanisms through which it's working. This is still to be worked out. One of the mechanisms is through sort of stabilizing the poly so that they don't degranulate as much and sort of elaborate the inflammatory response, and as a consequence, nociception. And also there may be a direct effect at the level of the periaqueductal gray in sort of inducing descending inhibition. But the whole idea is by using multiple drugs at the same time to control nociception, you get better control of nociception as well as unconsciousness. So we're actually starting a clinical trial on this at the BI hospital at Bala Subramanian to test this out in cardiac surgery. So let me just pass on to this last topic here, closed loop control of anesthesia and reanimation. So these are my colleagues, so Earl Miller, who I mentioned before, and Jake Donahue. This is work I'm going to show you in the non-human primates at Suresh Chakrabarty. Miriam Sinichu is one of my PhD students who's now a professor out at University of Southern California. And Shannon Cheng, who I mentioned before, is at WashU, an electrical engineering associate professor there. So these are all three well-known people who were placed in medical coma because of head injury. And, medical coma has been mentioned a lot as a term in the last few months or so in the wake of the COVID pandemic, because patients have been anesthetized for long periods of time. But that's probably a loose use of the term. But here, what this refers to is very specifically placing someone in a very profound state of anesthesia because you want to protect the brain. Either you want to stop seizures, which are intractable, or what you want to do is reduce intracranial swelling. And so what do? You dial up the propofol until you see this pattern. You saw this pattern appear in the lady that I did the induction on at the beginning of the lecture. And you see the burst and the suppression. So this is a profound state of the brain being turned off. You see it in hypothermia, you see it brains that are ischemic. It's actually showing up a lot in COVID patients now as well. But general anesthesia is one way you can systematically induce it. Now, you know you want this person to be in the state of burst suppression, but then the question is, how well do you maintain it? So what happens is you just decide you want them to be in burst suppression, the pump is started, the propofol is started at some sort of rate, or barbiturates is started at some sort of rate, and then someone walks away. So it's a very, not a very precise way to control the state of this brain state with the drug. It's like saying, look, I'm going to fly from here to Tokyo, and you point the plane in that direction, and every now and then you look to see if you're heading the right direction. You come back and turn the yoke one way or turn the yoke the other way. It doesn't make sense. If ever there were an opportunity to build a control system, this is it. So, and just to give you some sense, these are data from our hospital in which we had two groups of patients. So the blue patients down here are patients that had ischemic brain injuries, and the patients over here are patients that had epilepsy. And so we've color-coded these. So each column is a patient, and the colors correspond to whether or not they were in control, they were at the desired level of burst suppression. They'd be green. And if they're red, they're above the desired level. And if they're blue, they're below desired level. So this is data from our hospital. This is work done with Jen Ziang, who's one of my PhD students, and also Brian Westover in the neurology department at Mass General. And you can see a good part of the time for most of the subjects, they're below control or above control. And there's a very small period of time when they're in control. So again, speaking to so if you're targeting medical coma using medical coma as a way to sort of try to keep the brain in a state so you can reduce seizure frequency or reduce intracranial pressure, the therapeutic benefit's not being realized. So why not build a control system? You can imagine we tested this out in a rodent a few years ago. So the idea is you can build a controller where you have like a target level of burst suppression. And the cool thing about burst suppression is very easy to define what the target should be. You just say the fraction of time you want the system to be suppressed. So if there's no suppression, then it's 0. If it's totally suppressed, it's 1. And anywhere in between, there's a fraction of a number between, you can have a fraction between 0 and 1, the fraction of time the person is suppressed. So 0.5, 0.3, 0.2, whatever you want it to be, 0.7. So you set that level here, you estimate what it is based on the EEG, and in real time you change the infusion rate of the pump, and you just have this very simple sort of control system. But just to show you what this looks like, this is we did this a few years ago, so this is now 7 years old, just to show you what's sort of possible. So we're going to do this on a rodent, and we're going to start the green level here tells you the target, so it's 0.4. This is the EEG here. The EEG is it. And then this is the actual control signal. This is what the controller is doing. And here's how the pump is changing on a second-to-second basis to achieve this state. Now we're going to increase it. So we want it to be now 70% suppressed. So the pump dynamic changes dramatically. You can see here. And now you can see this is hugging right along the line of 0.7. And watch, the EEG is going to be coming down sort of more and more. And now we're going to take it up to 0.9. So suppress it even more, suppress it even further. But you can see it's tracking really well, the target of 0.9. The pump dynamics are quite different, though, in order to achieve this. And you can see the EEG is, it was up, came down, so it's much more suppressed here. So this is very easy to use. So this was over almost an hour. And so what I showed you was this animal here. So it's going up, going up, going up like this. And so it goes up, and when it comes down, excuse me, in order to come down, you will have a drug which can make it drop. So what happens is the controller turns the pump off for a period until you reach the level that you need to be at, and then it picks up again. So you can see going up, coming down, going up again, or all the various combinations we went through. This sort of control is very, very feasible. So, but in order to get something approved, we need to test it out in non-human primates, because improved for humans, we need to have a large animal model for it. So we've recently been doing these studies that Suresh has in non-human primates along with Earl Miller. And so one of the things that he figured out was, and you sort of saw this in the studies I showed you earlier, the power in this 20 to 30 hertz band declines very rapidly as the spiking activity goes. So we can't measure spiking activity, in the animals, or certainly in a human, we can measure EEG, and in the animals, we can measure field potential. So we're going to build a control signal out of this power band here. So let's say he's done that, okay? And he's set it up in such a way that it should be at 1. When the animal is well anesthetized, sufficiently unconscious, it's going to be at 1. So what he's going to do, he's going to start off with just letting the animal, letting the infusion rate run at a constant rate. And then we're going to let the controller take over. So I'll just play. Now the target is 1. Then we're going to decrease, then we're going to increase it. So you can see it started. And so the infusion rate is constant. You see in the middle panel there, the infusion rate is constant. And now what he's going to do is he's going to let the controller take over. And if you look here, see this is going down. Now the controller is taken over. And now it's right on target where it should be. So we'll hold it there for about 30 minutes or so. And now he drops it down. So make it even deeper. So it drops down to a lower level. And now what he's going to do is going to stay there for about 20, 30 minutes, and then he's going to bump it up to a higher level again. So this is controlling a state of anesthesia, which would be more like the state you'd want to have someone in for surgery, as opposed to burst suppression, where they'd be in the ICU for medical coma. So the point is that either state we think would be easily achieved, using these ideas. And so this is something which we want to refine further, so we can eventually test it out in humans. So one final thing, we anesthetize you, but we don't give you any drug, and we use drugs to do that, but we don't give you drugs to wake you up. And so I want to show you here these experiments, which my colleague Ken Salt has been doing for a number of years and has been refining. So this is an animal. This is an animal, of course, it's an animal. This is a rat. And he's what cats do with ADHD. They have to inject it in. Then he's going to flush it. Now we haven't turned the anesthesia off. Whatever is working, even though the anesthesia. Now he's got a turnover for this. You can see he's going to start much more movement. His rat's obviously like that fat. He has a foot moving there. So that's he's making effort. There you go. One more push. There you go. Yeah. So what's happening here? So Ritalin blocks the reuptake of dopamine. So this is this pathway starting the VTA, the ventral tegmental area, going through the limbic system here, amygdala, hippocampus, through the nucleus accumbens, and going on up to the frontal cortex. And so this is a pathway which is very important for cognition. People who self-administer Ritalin because they're in high-stress jobs or the stimulants like this report that their mind really sharpens up once they're on the drug. That's a dumb thing to do. I'm not recommending it. In fact, just quite the opposite. But the word on the street is this is something that works to help sharpen the mind. So if after anesthesia, people are having trouble coming to, or maybe just as a matter of course, to help people come to and improve their cognitive function after anesthesia, this is something which might be tractable. And this is different from, this pathway here is different from the diagonal striatal pathway here, which is the one implicated in Parkinson's disease. So I've shown you this clinically just by giving a rat Ritalin, but we've also done this in a very precise way, optogenetically stimulating the VTA and activating specifically the dopamine neurons to actually turn the brain back on. So this is very feasible. So this is something that Ken's been working on for the last few years, and he's about to start a new clinical trial work to test this out, as well as some other agents in humans. So I think this is something which should be part of standard practice in anesthesia. So I've just shown you that a primary mechanism, what we think that through which the drugs work, is anesthetics work, is by inducing oscillations. And I showed you directly in the monkey there that when those slow oscillations in particular are present, you have this amazing modulation of spiking activity. They're modulated and they're totally shut down across large parts of the cortex. So it's going to be very difficult for you to be unconscious when that's there. The practical implication of that is it's the same, the oscillations show up very readily in EEG. Remember, I said it's the strongest EEG signal around. It changes very systematic with the anesthetic, the age of the patient. And this allowed us to design control systems for both burst suppression as well as just sort of the standard anesthetic state. And I showed you a bit about multimodal anesthesia, the idea of using multiple drugs to target the nociceptive system. This is a much more efficient way to control nociception and pain. It makes control of unconsciousness also much more efficient as well. And it's an obvious way to move away from using just opioids to control pain during anesthesia or in the postoperative period. And then finally, reanimation, as I just showed you, turning the brain back on. What we do now is let it come back passively, and this might be one of the ways to help prevent or reduce the incidence of brain dysfunction, particularly in elderly patients. And so this is the way I view general anesthesia. It's this field of clinical neuroscience, which has all these very cool interactions with other problems. But we can only realize these interactions if we take anesthesia seriously from the standpoint of neuroscience. And, we can learn a lot about the brain by studying, general anesthesia, and also contribute to, studies in other areas. And I'll just give you one example, like ketamine is used to treat pain. Ketamine now in low dose is used to treat depression. And it's also a model for schizophrenia. I can give you a combination of drugs right now, which can write just like that, instantaneously create like a locked-in syndrome. Or like over here, the Ritalin that, we're using to help patients recover from anesthesia. In the rodent experiments we were using to induce recovery from anesthesia, our colleague Brian Edlow at Mass General is now using a paradigm based on that to help induce coma recovery. So all these things are possible if we take anesthesia more seriously and think of it as a subdiscipline of clinical neuroscience and not just a discipline of pharmacology. So I'll stop there. Thank you very much.
Thank you so much, Emery. That was really terrific. I just remind everyone, if you have questions, please put them into the give feedback section, and then I will try to ask as many of them as I can for you. We have a couple already. So one question that came in, Emery, was how important is it when you're thinking about anesthesia and dosing, et cetera, to know the patient's alcohol and drug use history? Does that affect the efficacy of anesthesia?
It affects it dramatically, and it's important to know. So for example, someone who's acutely intoxicated, very often it takes very, very little to anesthetize them. And so you can have, and then imagine someone who's has been a chronic, has a chronic drinking, but has an intact liver. Very often those patients might have a higher anesthetic requirement. And then once they've sort of gone over the edge, they have maybe a cirrhotic liver, and have profound hepatic dysfunction, then their anesthetic requirements may decrease. So knowing their drug usage history is important, and particularly like with drugs like with cocaine and what have you. And one of the things which we're really trying to sort out now is marijuana. Understanding what the interactions are with anesthesia. And right now that we don't really have a clear picture of it or how to think about it. So that's something which we're going to be sorting out in the next few years or so.
Great. So you have on this slide, you mentioned that ketamine is used, you're using anesthesia, but it's also being tested in depression.
Right.
Does your studies of ketamine give any insight into why it might affect intractable depression?
Not at the level that you're thinking about. It really doesn't. What we do know is that if I go back here to this, slide where I showed you the ketamine right here, this particular pattern right here. This is low dose ketamine. This is in the dose range. that people are getting when they when you give them when you treat them for depression. It's a very low dose. It's about, maybe somewhere around 30, 40 milligrams over like a half hour to 40 minutes or an hour or so. And this is the pattern that you see. So this tells you, so this is what we would see in the operating room if we gave a similar dose. But the part that's hard to explain is, why does this effect persist? And so that's the part, that's, if you would, the $64,000 question. I think one of the things to also take away from that is that if ketamine is persisting like this, other drugs are persisting as well, so that the effects that you see post-operatively really do represent, these sort of, just because the anesthesia is over, you've turned it off, there are these effects that have been happening in the circuits which just don't resolve just because, the person's out of the operating room, has been extubated. So this is an open question for us right here, why this occurs, but it reminds us that the side effects of the other drugs are probably there as well for long periods of time.
Yeah, and actually one of the questions that just came in is related to that. Are multimodal, multi-drug anesthetics appreciably susceptible to adverse reactions, specifically from incompatibility of drug combos? So does that create a problem?
No, there's not the incompatibility in the sense that I think the questioner is asking. And in fact, what you actually see is more compatibility, I would argue, because what we found empirically is that when you push one system too much, like when you overuse, let's say when you overuse the opioids here, people used to say, keep it simple. All right. Well, this is actually one of the situations where actually more is better. And more doesn't mean you're giving large doses. You're actually using, reasonable doses, or like in many cases, lower doses, but it's the synergy that you're taking the effect of. You're controlling nociception through here, through here, through here. And actually they're hitting different parts of the nociceptive system. I'm not going to go so far as to say they're orthogonal parts, but they're certainly different parts. And again, the payoff is that you get unconsciousness for that as well. So the propofol, you're able to reduce the dose and get a set unconsciousness at a much, much lower dose. I was just showing that later. I was giving 30 mics per kilo per minute, which is literally unheard of.
Incredible. So another question about the effects of anesthetic response in the aging brain, which are fascinating. Do these correlate in any way with cognitive function or other neurological defects? Have you looked at that?
Well, that's something we're going to start. Actually, my colleague, Patrick Purdon, is actually looking at this question right now, even as we speak. I mean, I've had a lot of anecdotal experiences. Like, I had this gentleman who was 77, and he was a retired accountant, financial analyst. And his EEG looked like this 30-year-old. And by the same token, I was taking care of this young guy who was like 30, in his 30s. And I couldn't put my finger on it. Something didn't seem quite right, just in the conversation. And then I always ask my patients, do you do sports? What sort of athletics are you involved in? He goes, yeah, I run now. I says, but I used to play rugby. All right. And, when I got him into the OR, I looked at his EEG looked like this 57-year-old guy here. So there's something there, and, whether it's something we can actually then codify and use systematically, either for, we certainly can use it for anesthetic purposes, because once you see those sorts of patterns, it's pretty clear to us, that my experience is that I don't need as much anesthesia to anesthetize those patients. But could it be used as a diagnostic tool, kind of the way I've suggested here, like in quotation marks, old brain, young brain? I think the jury is out on that, but it's certainly like a tantalizing possibility.
Yeah. We have time for maybe 2 more questions. We've got a ton of questions, so we won't get to them all. Do you think Ritalin might be useful to treat long-term brain fog, for instance, after concussion or, COVID-19 long hauler syndrome?
I mean, that's a very interesting question. In fact, my colleague, Mika, there's a group of us, a consortium of us between Columbia, Mass General, and Cornell, who are actually looking at that very question. It won't be simple as a simple drug therapy, because there are a couple of components to it. I mean, one of them is probably, many of those patients were on anesthesia for a long period of time. And the other effect is they probably had periods of hypoxia, which, certainly contributed to this brain fog that, persists afterwards. It's certainly a reasonable idea as a therapeutic option, I think, probably to start with. But I think we have to realize that the actual genesis of that brain fog is probably multifactorial.
Okay. All right, one more question out of many we could choose from, but does your multimodal approach accommodate for individuals who cannot tolerate gas anesthesia due to adverse effects?
Oh, exactly. I'm glad you asked that. So I stopped using gases 4 years ago. In fact, all the drugs that I'm looking at here are intravenous drugs. So, my colleague, Marusa Naranjo, Dr. Naranjo in Mexico, she uses just a little background of the gases, but I pretty much stopped using them because I found that I can titrate much more effectively the state of unconsciousness using propofol than I can with the gases. So it gives us a way to move away from the gases, actually, which some we're going to have to do anyway because they're pollutants.
Huh. Just absolutely fascinating stuff, Emory. I wish we had more time for all the questions. Hopefully some of these people will reach out to you directly with these great questions. And I'm sorry we had to do this virtually. Normally we would have a time for tea and cookies in the library, but we'll have to do that another time.
Okay,
we can have our own tea and cookies in our houses.
Very good.
Thank you so much, and thanks for everyone for joining.
Great.
This is terrific.
Great, thank you. Thanks for having me.